Welcome to our talk on Porifera and Placozoa. This is the first category of true animals that we're going to talk about in this class, and these are covered in chapter 12 of your textbook. So Porifera, or what we also call sponges, so I'll use that term interchangeably. Sponges are the sister taxa of all animals, and so they're kind of the most base plane, first kind of branching off when we're looking at the taxonomy of animals. And they represent a cellular stage of complexity. So they're composed of a whole bunch of different individual cells that are associated with each other through a extracellular matrix, but each cell is still independent. And they have very simple morphology, so they're not complicated animals to follow. They do have various different shapes and things like that, but they're pretty simple. Most are asymmetrical, but some can be radial. And if you look at many pictures of sponges on the internet, you'll see kind of what I mean, that they come in a variety of different shapes and things that don't really have a defined symmetry. All sponges are sessile, but they do have a free-swimming embryo, and they have no mouth and no digestive tract. So how do they feed? Well, through suspension feeding, and we'll go into far more detail on that in later slides. Some capture prey, but the vast majority of them undergo suspension feeding. Like I mentioned, they can vary in shape, but they can also vary in size and color. So you can have sponges that look more like a mat of moss almost on something, but that's actually a sponge. You can have some that have this large tubular shape, like you see in this picture here. Some can look like a fan. They have all types of morphology, and some can be tall, like meters tall, and some can be very, very, very thin and small, and they can have various colors, right? So there's a lot of different diversity out there, and some of the prettiest pictures to look at of the marine environment are of sea sponges. And most of them are marine, but you can have sponges that live in freshwater environments and in brackish water, and brackish water is where fresh and salt water meet, but the vast majority of them are marine. And when we look at sponges, and we also look at these other organisms that we talked about in the last lecture called choanoflagellates, we really look at them to kind of get an idea of how animals came to be. How did we get the tissues that we have that are multicellular, and how are we organized into multicellular organisms? So sponges, even though they're simple and you might think about them in that way, they can give us major insights into how we ended up with the vast majority of kind of multicellular uh, animals that we have on this planet. So before we get into sponges really heavy, I want to give you guys some more details about the choanoflagellates. And so just to reiterate, the choanoflagellates are not sponges. They're not considered animals. They are protozoans, but they are a sister taxa to animals, and they are also aquatic. And this is actually a picture of what a choanoflagellate looks like. They can live alone, or they can live in colonies. And so just as a reminder from like last talk we talked about, when you have a colonial lifestyle, they're different cells that kind of aggregate together, but they don't differentiate. So they're not a tissue. They just are individual cells that just live together, right? So it's like having you and your roommates versus you and your family, right? You just live together. And they have this flagella, which you can see in this picture here. And unlike what you would normally think of a flagella for locomotion, in this case, choanoflagellates actually use their flagella to acquire food. So they're mostly sessile, and they stick to a surface, and then they use their flagella to whip the water around them so they can eat. They highly resemble the choanocytes that we see in sponges. So this image here is actually the choanocytes from sponges. And just looking at them, you can tell, you can see the resemblance between the two types of cells. And when you're looking at choanoflagellates, one interesting thing about them is that they have cell-to-cell communication, and they kind of can adhere to surfaces and to each other in ways that are very similar to what we see in animals. So that's kind of how we end up looking at them and sponges to try to kind of get an idea of how we ended up with multicellular animals. In addition to looking at choanoflagellates to get an idea of maybe how multicellularity evolved, we can also look at sponges and to see how epithelial tissue may have evolved in higher order animals. And so in order for a tissue to be considered a true epithelial tissue, it must have two characteristics. There must be these junctions here that connect and anchor neighboring cells to one another, and these are called desmosomes. So desmosomes must be present, but also you must have these anchors here that anchor the cells to the basal lamina. So in order for epithelial tissue to be considered a true tissue, it must have both of these components. In sponges, we don't necessarily see true tissues, and this is because in some species they'll have one of the two of these characteristics, and in other species they might have another. So, for example, placozomes have been found to have desmosomes, so they have those cell-to-cell anchors, but they don't have a basal lamina. So we haven't observed a basal lamina yet in those organisms. On the opposite side, some species of sponges have a basal lamina and cells are anchored to it, but they lack desmosomes. So these are not considered to be true tissues, but when you're looking at them, they could give some idea of how tissues evolved over time in higher order animals. So you can also look at these tissues, these non-true tissues, to identify how cell-to-cell communication and cell-to-cell adhesion may have began, because they're homologous in sponges to other animal taxa. You can also look at these tissues to identify the more complex regulatory pathways and how they evolved over time. And this is because even in sponges, they exhibit anterior-posterior axes in their larvae, even though they're very, very simple and they don't have a whole bunch of other axes, they do exhibit anterior-posterior axes. And then if we're looking at the sponge larva, and then we kind of look at other organisms over time, more and more complex organisms, we might be able to get some idea of how these complex regulatory pathways kind of began. So porifera, or sponges, have some core structure. So they don't have necessarily a true skeleton or anything like that, but they do have these fibers that are called spicules, and they can be made of silica or they can be made of calcium carbonate, and they provide some sort of structure to the sponge. Otherwise, they would probably be like a jelly filled just glop, right? So it's not necessarily a true skeleton, but it does provide some structure. And the types of spicules that are formed are one way that taxonomists go about classifying different sponges, and in addition to the chemical composition of that sponge. So this image down here shows you some different spicule formations that can occur in different phyla of sponges. And then also it's been found that sponges that use calcium carbonate to form their spicules form a whole different clade than those that use silica to make their spicules. And so it's actually very interesting to see that you can use genetic analysis to definitively say these two types of sponges are in a different clade from one another. And it's been hypothesized that maybe there was an ancestor, and they found some fossil evidence of an ancestor that had both calcium carbonate and silica in their spicules. And so that may be another puzzle to how we ended up with the sponges we have now, and the evolutionary history of sponges. Sponges can also harbor microalgae and cyanobacteria in their tissues and on their body. And a lot of times they can engage in a mutualistic relationship with these microorganisms that reside in and on their tissues. So sponges have a variety of cells that are associated with one another that allow the organism to survive. So like I said, these are at the cellular level of complexity, so these cells do not form tissues, but they are kind of interacting with one another and kind of held in close proximity to one another via the extracellular matrix. And some of the cells that are involved in making up a sponge are mesophyll. So the mesophyll is the connective tissue that houses all the different cells that are that make up a sponge, and they can also be involved in respiration and excretion of waste via simple diffusion. And then we have our choanocytes, which are here in this image, and we'll talk in a lot more detail about those in the next slide when we talk about feeding. We have our archaeotypes, which is like this pink cell here. And these are amoeboid cells that live within the mesophyll, and they travel back and forth throughout the mesophyll. And they are responsible for helping with digestion of food and transport of nutrients to cells throughout the sponge that need it. But they also are pretty dynamic, so they can turn into whatever type of cell that the sponge needs. So they can function in various different ways. So they're very important for a sponge. And then we have our pinacocytes, which are in this image these purple cells. And this is the closest thing that a sponge has to an epidermis. These are their dermal cells that kind of lie on the outside of the sponge, and they can also engage in food acquisition through diffusion and active transport. So one interesting thing about sponges is they're very, very simple, but they can contract, they can move. They have pores on their surface, and they can make those pores smaller or bigger. Some of them have an ocula at the very, very top, so they're kind of shaped almost like a bowl, and this would be the ocula. And some of them can expand and contract that ocula. Now, these movements are slow, very slow, but they're possible. And it's really interesting to scientists of how is movement possible when there are no tissues. There's no nerve tissue, and they're just a bundle of cells that are associated with one another. And how does this occur is something that scientists are really looking into to figure out how do you get coordinated movements without a nervous system. Porifera are filter feeders, and so they're a primary consumer that feeds on particulate matter that just is floating in the water column. And a lot of this can be, small cyanobacteria, it can be plankton, anything, detritus, just small organic matter that's suspended in the water column is consumed by sponges. And they are able to eat using these choanocytes. So this down here, this image is showing you kind of a pictorial drawing of what a choanocyte looks like. And the job of a choanocyte is to bring in water and oxygen, or bring in food and oxygen from the environment, and then to remove waste. They can also play a role in reproduction as well. So how is the process of eating done for a sponge? So essentially, this choanocyte has a flagella, and it'll whip its flagella around, which will cause movement of water into the pores of the sponge and through canals. So in this image, you can see water is coming through from some pore on the outside surface of the sponge, and the water is coming into these canals. And in this case, there's a kind of channel that's lined with choanocytes. In some sponges, this is how the process works. In some sponges, the choanocytes kind of line the inside near where the ocula is, but just in general, we're just going to use this as our general example. So the kind of the bulby part of the choanocyte is lodged within the tissue, well, not tissue, but lodged within the mesophyll of the sponge. And then the flagella is on the outside. And like I mentioned, it's whipping around, which causes water to move into the channel. When water moves into the channel, then we'll go down to this image below. The water will then go into past the choanocyte, and it has like this sieve. So you can see that there are microvilli that make up this mesh kind of sieve part of the choanocyte. And this allows them to capture food. So very, very small particles can go through the sieve and will go down to the kind of this basal body portion of the choanocyte to be phagocytosed. Larger food particles can't pass through that sieve, but they'll just basically run down the side to the basal body part, and then from there they will be phagocytosed. So both very, very small particles and slightly larger particles can be phagocytosed by choanocytes. Very, very large particles will never even make it to the choanocytes because the pores on the sponge are very small, right? So when I say big, they're big in comparison to the kind of this sieve-like structure of the choanocytes via the microvilli collar, but they're not big as in like actual huge pieces of detritus. And so essentially the food particle will be phagocytosed and will start being broken down by the choanocyte, and then the choanocyte will pass on the partially digested food products to the archaeotype, which is shown here in this picture, and then the which are, remember, those amoeboid cells will then go ahead and digest the food even more and transport it all throughout the sponge to cells that need it. So this archaeotype can also get food from the pinacocytes and do the same type of thing as it's involved with the choanocytes as well. There are other cells that are involved in feeding, like we talked about. You can have diffusion in and out, the pinacocytes, phagocytosis, and things like that. They can absorb nutrients from the environment through simple diffusion. So there are different ways, but really knowing this process of how choanocytes are used to obtain nutrients is important. Porifera can reproduce using sexual or asexual reproduction. In asexual reproduction, they can use fragmentation. So this is where you have a part of the sponge breaks off, and then if it's the conditions are right and it's met with a substratum, it can form its own new sponge. And you can also take a sponge, and if you kind of break it up into fragment it into parts and put those parts close to each other, they can actually start to reform or regenerate that initial sponge. So fragmentation is one method. We also have budding, and budding is where you have an offspring that offshoots from the parent. And in this image here, it's showing you there's a bud on this sponge, and that bud will then be disassociated, and it can travel through the water column via currents and things like that to this another substratum, and once it anchors itself, it will form a sponge, another sponge. And this is, a form of cloning, so these individuals are identical. And then you can have also gemulation. So in gemulation, a gemula is formed, and this image is what that is. So gemula. And a gemula is essentially almost like a spore for sponges. And this is common in like freshwater sponges that are in temperate areas that experience different seasons. So in the winter, it's cold, there's less sunlight, so these sponges will die. And when those sponges die, they leave behind these gemula, and they will basically keep all of the components required for that sponge to regrow kind of safe and dormant throughout the winter And then once conditions become right, that gemmula will form the sponge again. So think about it as a spore for sponges. And then we also have sexual reproduction. So sexual reproduction can occur through gametes from two different sponges, so two different organisms that are kind of near each other, or it can be through a monoecious method. And so monoecious method is when the sperm and egg both originate from the same individual. And in this case, we'll follow this. So the choanocytes or the archaeocytes can become sperm and egg depending on the species. So I mentioned choanocytes can play a role in reproduction. This is how. So the choanocytes or the archaeocytes will become the sperm or egg, and the egg will meet with the sperm and give you fertilization. So after fertilization, start undergoing cell division and to give us an embryo. So our embryo now is kind of not multicellular, but the embryo consists of multiple cells, and then we have our which is all going to be ectoderm, remember, and then we have our choanocytes, and then the arrangement of the embryo will form into something that looks kind of like this, which is our larval stage. And the larval stage is motile, it's ciliated, and it can move throughout the water column. So in addition to it being motile, if you have the right current pushing in the right direction, you can actually move this larva to a satellite location where it will then interact with the substratum and form a new sponge. In this image, it also didn't go very far, but due to currents and due to being motile, they can go to various areas. And then this act of sexual reproduction can be viviparous or oviparous. So in viviparous, the sponge will keep the embryo within itself for a period of time, and then oviparous is like what we just talked about, where the embryo is released into the environment and goes off to establish somewhere else. Sponges, as we mentioned way back in the early development PowerPoint, they stop at the blastula stage of development. So they do not undergo gastrulation or anything like that. They only have one tissue layer, which is ectoderm. And some embryos have been found to actually have two germ layers, but at some point those two germ layers are lost, and you end up with just a single germ layer of ectoderm in the adult stage. So the last topic we're going to address are Placozoa, and there's not very much known about this phylum of organisms. They were recently studied and classified as their own phylum. They're very small taxa, but some interesting things about them is they have some of the smallest nuclear genomes of all animals and some of the largest mitochondrial genomes of all animals. So that kind of makes them distinct and kind of sets them apart. They have a plate-like body, so this little pancake thing is supposed to show you kind of how flat they are. And they they're considered to be diploblastic because they have kind of two germ layers. They have their dorsal epithelia, which is what's being shown to you here, but it's also in this image up kind of like the back part that you would see if you were looking at it. And they have the ventral epithelia, which are these cells here. And that's the part that kind of is on the sea floor or on the ocean bottom. And the ventral epithelia is what's responsible for feeding. And so these organisms feed via absorption. So the ventral epithelial cells will extrude enzymes that will break down their prey, and then they just basically absorb the nutrients from that digestion, the external digestion that occurs. And it's been found that they reproduce asexually. There hasn't been any observation of sexual reproduction yet, but however, studies into Placozoa are relatively new, and so with time we might find that these organisms can undergo sexual reproduction, but at this point, there's really only evidence for asexual reproduction. Okay, so now we are finished talking about sponges. Just remember some key things about sponges, their morphology, how they feed, ways that they can reproduce, things like that. Go ahead and read chapter 12, 12.1, section 12.2. Just make sure you get the big picture, like I just mentioned about the morphology of sponges, what are spicules, what are choanocytes, what are archaeotypes, how do they go about feeding, how can they reproduce, things like that. And then read section 12.3, which is on Placozoa, and then also do the smart book assignment for chapter 12. Excellent. See you in the next talk about jellies.